All right, so now we go with the second speaker, Ulf Smith, and I'll do a brief summary of Ulf's achievements. Ulf is professor of internal medicine and director of the Lundberg Laboratory for Diabetes Research at the Sahlgrenska Academy at the University of Gothenburg in Sweden. He was former president of the European Association for the Study of Diabetes, chair of the European Foundation for the Study of Diabetes. He also was past president of the Alliance for Biomedical Research in Europe, and he is chairman and member of several scientific advisory boards, and he has current position as a member of the European Commission Scientific Panel for Health at the European Commission. So based on that, it is more than clear that he is one of the most influential people in diabetes research in the world. In addition, he's still doing extremely good research, focused essentially on the pathogenesis of type 2 diabetes and complications. And he's done many different things, co-author of more than 500 papers, but he has done, or at least from my point of view, something that I think is absolutely phenomenal is the work he's done, demonstrated that adipose cell hypertrophy is a driver in the pathogenesis of type 2 diabetes. And I think this is one of the fundamental observations he's contributed to diabetes. In any case, it's a luxury to have Ulf here, so whatever you want.
Thank you very much for this very kind introduction. Okay, so I will take a very different stand than Jorge did. I'm going to talk about translational diabetes research more from a clinical perspective, because that's how I understood that this symposium was supposed to be run, considering the involvement of MSD. So if I will please interrupt me if I am not clear in terms of, well, if you don't understand, interrupt me. Okay, so with this topic, I could basically talk about anything, translational diabetes research, because if it's translational, it's supposed to benefit patients and society. And you can look at how the NIH has phrased this, because they have established what they call the National Center for Advancing Translational Sciences, NCATS, and it's one of the institutes and centers at the NIH. And it is supposed to conduct, support research on the science and operation of translation to allow more treatments and get more patients, and two patients more quickly. And the center focuses on what's common in terms of a translational process. But the idea is, and I think it's a great idea, and I would actually hope that the European Commission will also try to establish core facility with a focus of translating science to the benefit of patients, because, well, I don't want to be political, but political world views a lot of basic science developments as being more for the glory of the scientist than for the benefit of society. And we need to have tools to pick up on interesting developments, and maybe a translational center like the NCATS is something that Europe also should consider. So basically, translation is turning observations in the laboratory, clinic, and community interventions that improve the health of individuals and the public. And of course, the politicians are very interested in making sure that science reaches patients, i.e. is translational. Okay, so basically, I mean, there are so many things which are part of a translational diabetes research. It is new knowledge translated to reach patients with diabetes. I could talk about technological developments in diabetes therapy and drugs, etc., which are important for the patients. Type 1 diabetes is an autoimmune disease leading to the loss of beta cells and with specific problems. Type 2 diabetes, insulin resistance, mainly due to obesity and dysfunction of beta cells. And of course, the very costly and for the individual suffering, the long-term consequences in terms of the diabetic complications. So all of these that we are working in needs to be translated so it really reaches the benefit of patients. That's the idea with translational diabetes research. And I'm interested in understanding why obesity drives type 2 diabetes, or is associated with the development of type 2 diabetes, to be precise. And what's happening in the world today is that we have this obesity epidemic, which is obesity due to changing lifestyle in different ways, driving the type 2 diabetes epidemic. And this epidemiologist really reviewed what's happening, says that whatever prediction made by WHO or other formal institutions or bodies have underestimated the diabetes epidemic. Currently, we believe, at least 2015, there were 415 million patients with diabetes, and it's predicted that in 15 years we're going to have over 500 million. So it's a huge challenge for patients and for society, and we really need to have new tools to identify risk, to prevent development, and to treat the diseases. Okay, and this is global, and this was the 387 million was the forecast by IDF, I believe, and is now 450 million. And it is throughout the world, very much so in Asia and China, and of course in the US. And the Arab world has still the highest prevalence of diabetes in some of the countries. Okay, so it is driven by obesity, there is no doubt, and lifestyle factors, and the genes have certainly not changed over this very short period of time. It is your lifestyle, and it is the development of obesity, which is the major driver of type 2 diabetes. And obesity is not just type 2 diabetes. It has a lot of other consequences, and in fact, it is obesity and overnutrition is the second largest cause of premature death in the world, except in sub-Saharan Africa. But in the rest of the world, it is really because of these associations. It's not just a question of type 2. It's also associated with other common diseases, cardiovascular disease and its risk factors. NAFLD, do you know what NASH is? NAFLD is the non-alcoholic fatty liver disease, which is associated with obesity, associated with cardiovascular disease, and driven by obesity. And it's predicted that it's going to be the major cause for liver transplantations in the US in 10 years' time. So it is increasing, it drives cirrhosis and liver failure. Cancer is also part of the obesity epidemic and risk, and that is, I'm just sort of showing a paper from the Lancet Oncology, where they estimated that about 3.6% of all new cases in adults were attributable to high BMI. So it is also a problem for cancer. And we have also learned that it is a question and a problem that diabetes increases the risk of dementia. Not just the vascular dementia, because we all know that diabetes is associated with increased risk of stroke and small emboli, which lead to dementia without having the manifest stroke. But it's also now a lot of evidence that it is also a risk for the non-vascular dementia, i.e. Alzheimer's disease. And that is in fact also a problem for metabolic syndrome and obesity. So it has huge impact on health and common diseases in society today. And I won't go through this, but just sort of emphasize that diabetes and hypoglycemia and frequent hypoglycemia is associated with increased risk of cognitive disorders or dementia. But also insulin resistance in certain areas of the brain are also found to be important in terms of the effect of insulin in actually activating insulin receptors and signals in the brain. And there is a lot of work ongoing, hoping that maybe even nasal insulin therapy will have a protective effect on the development of cognitive disorders. And there is a pretty big study currently ongoing in the US and funded by the NIH, where individuals are treated with nasal insulin and following cognition to see if it sort of limits the slope of development of cognitive dysfunction. Okay, we've heard about genetic risk markers for obesity, for type 1. And the type 1 is really an HLA, it's an autoimmune disease. Obesity, the genetic risk markers we have are primarily genes that are functional in the brain and regulating food intake. The last number I heard from, I think, Marco Laxo said that it's been described up to 180 associations for genetic markers associated with type 2. I don't know what is true. Jorge says 100, and it's probably at least 100, let's put it that way. But these DNA polymorphisms can only explain a pretty small part, and I would think that Jorge's presentation was a very interesting eye-opener for what's going on and may indeed be an explanation for the fact that type 2 diabetes is clearly a hereditary disease in terms of family history increasing the risk of developing type 2 diabetes. If you have a first-degree relative with type 2 diabetes, you have 30 or 40% lifetime risk of developing type 2. If you have 2 first-degree relatives, you have about 60 to 80% risk of developing this polygenic disease type 2. So there is a strong influence by heredity and genetics, but the DNA polymorphisms have not really identified what these risks, where these risks are and which genes are involved. But I should also add that obesity is associated with type 2 diabetes and insulin resistance, even without any genetic risk markers. So we have a lot of things to learn, and I think the approach that Jorge and his group and probably others are taking is really very, very interesting and may open up our eyes for how genes are regulated and lead to the development of type 2 diabetes. So that is sort of the short background clinical overview of obesity and particularly type 2 diabetes. And obesity obviously involves the adipose tissue. One question, which is a translational approach that is being made currently in both pharmaceutical industry and also in university laboratories, is to understand, can we get these white fat cells that many of us have too many of, or at least are too large, can we get them not only to store fat, but can we get them to burn fat, to oxidize fat? And can we turn the white fat cells into oxidative beige brown? I think the brown adipose tissue in man is really too small to be a good target. But if we can get the large white adipose cells to become oxidative, that could be a very interesting approach. Skeletal muscles have been tried with different targets, and it's been very tricky to get the skeletal muscles to increase the oxidation. So there's a lot of focus now on the large white adipose tissue to see if that can become oxidative, increased mitochondrial biogenesis, etc. And there are some interesting results and studies ongoing. Okay. Then we have the typical Caucasian. This is not how a typical Asian, Japanese, Indian, or Chinese for that matter, most Chinese, look. But in the US, I guess, and increasingly so in Europe, this is the typical distribution of the adipose tissue, what we call the abdominal distribution. So it's not only the amount of fat, it's also how it is positioned and distributed. And the abdominal distribution is associated with an increased risk. Then you can raise the question, is this just a marker? So we do a waist circumference and say, well, you have over 110, so you have a high risk of developing type 2 diabetes. But is it actually causally important? And this is where genetics can be useful for this particular question. And there is a recent study, and I won't go through it in detail, but what they did was to look at genes which are associated with an increased waist-hip ratio, i.e. increased waist distribution or abdominal distribution of the fat. And then they adjusted for obesity, BMI. And see, is there, when we use this approach with genetic risk for having an abdominal obesity. So I think this is a very common approach where you basically try to use a Mendelian randomization approach. See, is this causal or is it non-causal? Is it just a marker or is it actually potentially causal? And I think that the conclusion from this was very clear. A genetic predisposition for this abdominal distribution of fat is associated with an increased risk of both type 2 and coronary heart disease. And it actually supports what we have been proposing for a long time, that there is a causal association between abdominal obesity, type 2 diabetes, and cardiovascular disease. And this is just shown here using a polygenic risk score for abdominal distribution. And as you can see, well, hopefully you can see it. There is, it's about a twofold increased risk of developing diabetes, irrespective of BMI, with an abdominal adipose tissue distribution, and also cardiovascular disease. So it's more than just a risk marker. Okay, let me then turn to something different. I would talk about two things which are more scientific and things that we have been doing. If the adipose tissue plays a role for insulin resistance and type 2 diabetes through obesity, the question is the adipose tissue important for glucose disposal following a meal or whatever? And the answer is no, it is not. It's not very important. It only accounts for about 10% of a postprandial glucose load. The rest goes to muscle and some to liver. And we were quite surprised when we did this study where we quantified the glucose transporters in the adipose tissue. And then we related that to effect of insulin with a euglycemic clamp, which is what this glucose disposal rate means. And we found a very nice correlation between the number of glucose transporting proteins and insulin sensitivity. How can that be when the adipose tissue accounts for about 10% of the postprandial glucose uptake? And this is really work that Barbara Kahn did with her animals, where she also showed that if you silence GLUT4 in the adipose tissue, the animals were about as insulin resistant as if you silence GLUT4 in skeletal muscle, which accounts for about 85% of glucose uptake, insulin-stimulated glucose uptake. And if you overexpress GLUT4 in the adipose tissue, you see an enhanced glucose tolerance and actually protecting the mice from the negative effects of obesity. So this small tissue is very much dependent on glucose uptake and metabolism. And the question is why? And then this is work that Barbara Kahn has done and Alan Sagatilian when he was at Harvard. And what they did was to use these mice and the adipose tissue from the mice and looking at what sort of lipids are secreted by the adipose tissue. And a lot were known, but they found this novel lipid family up on the right with the, you can see it's 16 to 18-fold elevation of these markers, and which actually had a structure which was not known before, but secreted by the adipose tissue. And it is called they're actually esters of two fatty acids, so they're hydroxy fatty acids. So they're called the fatty acid, hydroxy fatty acid of FAFAs. And there are quite a few different, but one of the most prevalent one and common one is the palmitic hydroxystearic acid. And they're actually shown not only to have an anti-inflammatory effect, increase the effect of insulin on glucose transport, but also to have some positive effects on insulin secretion. So we measured these fatty acids, and this is a very busy slide, and I apologize for showing it. But at least it shows the point that when we measure the circulating levels of the adipose tissue levels of these novel fatty acids, we see clear correlation with glucose disposal rate, I think that is up here, and also difference in terms of insulin-resistant adipose tissue versus insulin-sensitive adipose tissue. And this is now a big ongoing study in Barbara Kahn's lab, and we are collaborating on some aspects to identify the formation, which enzymes are involved, how is it cleared, can it be a new translational therapeutic possibility? We'll see. So at least that was a translation of basic science and into potentially of benefit for patients. Let me then end with something which we are quite puzzled by, but have started to study quite intensely in the lab. It's important to identify early risk markers of type 2 diabetes. Can we have easy markers, just taking a blood sample or plasma, and find markers which are associated with, causally, or just being a biomarker of risk of type 2 diabetes? And can we then initiate early therapy? So in an EU study that I'm involved in, we started off by doing what we call an untargeted metabolomics, i.e. measuring 1,200 small molecules in blood or in serum. And then we did that in very carefully phenotyped individuals, same BMI with and without insulin resistance, obese with and without insulin resistance, and a few additional. And when we analyze this small molecule, mannose, which has been known for at least 30 years to be important for some rare genetic diseases. But it's important for O-link glycosylation of some proteins. But not very much more has been known about it. It's a circulating metabolite, basically. And we found it to be a very good marker, and I will show some data from this. We also did, as part of a systems biology approach, looking at pathways when we found that mannose was increased, looking at some of the, at that time, known pathways for mannose formation. And we found that they were upregulated in obesity in the liver. Interesting, potentially interesting. So this just shows that mannose was one of the highest differentially circulating levels in plasma from individuals with and without insulin resistance. And that was independent of BMI because we adjusted for BMI. So that was kind of interesting. But then we asked a more critical question. Is actually mannose levels at baseline associated with future risk of type 2 diabetes, cardiovascular disease, and even complications of diabetes, such as renal complications and albuminuria? And the answer is yes, it is. So this is just the association of mannose levels and incident type 2 diabetes in two large cohorts. METSIM is about 10,000. EPIC Norfolk with Niqueram is about 300,000, if I remember correctly. So we collaborated with Marco Laxo in Finland and Niqueram and his group at Cambridge. And the answer is, even when we adjust for all potential confounders, including family history, including lifestyle, including smoking, including BMI, and also for circulating glucose levels, different markers, FPG is fasting plasma glucose, and 1,5 and hydroglutatol is something which also reflects glucose levels. And they did not remove the increased hazards ratio, HR, here, in terms of future risk of type 2 diabetes. Very puzzling. Not only that, it actually also turned out to be associated with one of the really severe problems with type 2 diabetes, heart failure. And that is a very high mortality. If you develop heart failure with type 2 diabetes or diabetes, your survival time is usually very limited, less than a year, if it's severe heart failure. So even in terms of these cardiovascular complications, we saw increased risk associated with baseline mannose levels. Even when we used a genetic approach to look at individuals who have increased gene markers associated with elevated glucose, insulin, or impaired insulin secretion. We also found that there is an association with elevated mannose levels. So individuals with a genetic risk for diabetes, and these identified markers here, have elevated mannose levels. And even more puzzling, when you look at progression of albuminuria as something which is one of the major complications in diabetes, that was also associated with increased mannose levels. And mannose is actually excreted via the urine. And that was true in type 1 and type 2 diabetes. And now we are extremely puzzled and have initiated a clinical study where we are giving mannose to elderly individuals and doing a very careful phenotyping to see, is there a direct effect that we can identify? And interestingly, there's also recent data that mannose is associated with TGF-beta signaling, which is a well-known risk factor for renal disease. We don't understand it, but it's something that we are working on, and hopefully we'll find pathways and mechanisms, or it may end up with nothing. My belief is that it will end up with more than nothing, and it may actually have a pathophysiological contribution to the development of diabetes and or its complications. I just wanted to bring up this as sort of a translational approach trying to identify high-risk individuals at an early stage and hopefully contribute to intervention and, or for that matter, if the pathways are important, as a new therapeutic target. And it's absolutely true what they say at the NIH in the NCATs. Translation is a team sport. It is not for one small group of individuals. You really need to collaborate. And I think that Jorge's acknowledgement list also identify how important it is to collaborate with different expertise, availability of patient cohorts that have been carefully phenotyped genetically or whatever. So with that, I thank you and willing to take whatever questions you may have.